Good morning, friends. This is Dr. Kanitha, serving as Assistant Professor and Head, Department of Biochemistry, JJ College of Arts and Science, Autonomous, Pudukkottai. In this video, we are going to discuss about monoclonal antibodies. Monoclonal antibodies are the antibodies which are identical because they have been produced by one type of immune cell. That is, all clones have been derived from a single parent cell. Whereas the polyclonal antibodies represent the antibodies which are being produced from multiple clones of B lymphocytes. Therefore, they bind to a number of different types of epitopes. There are various differences between polyclonal and monoclonal antibodies. Polyclonal antibodies are derived from different B lymphocyte cell lines, whereas monoclonal antibodies are derived from a single B cell clone. Polyclonal antibodies they vary from batch to batch, affecting the antibody reactivity and titer, whereas the monoclonal antibody offer reproducibility, predictability, and potential inexhaustible supply of antibodies with exquisite specificity. Polyclonal antibodies are not that powerful tools when it comes to clinical diagnostic tests, whereas monoclonal antibodies enables the development of secure immunoassay systems. This monoclonal antibody, otherwise known as magic bullet, was first proposed by Paul Ehrlich, who at the beginning of the 20th century postulated that if a compound could be made that selectively targeted a disease-causing organism, then a toxin of that organism could be delivered along with the agent of selectivity. This led to the production of humanized monoclonal antibodies, which helped to combat the effects of various other monoclonal antibodies in the previous years. The hybridoma technology involves the fusion of myeloma cell lines, which are immortal in nature, to that of the B lymphocytes, which are involved in the production of antibodies. The basic principle behind the hybridoma technology involves the immortal nature of myeloma cell lines. Also, the myeloma cells they have lost the ability to synthesize the hypoxanthine guanine phosphoribosyl transferase enzyme, which is being used up via the salvage pathway for the biosynthesis of nucleic acids. This salvage pathway actually it enables the cells to synthesize the purines whenever the major pathway gets hindered up. The selective culture medium is known as HAT medium, namely hypoxanthine, aminopterin, and thymidine. The unfused myeloma cells are unable to grow in HAT medium because they lack HGPRTase enzyme. Similarly, the unfused normal spleen cells also will not be able to